So, good afternoon, everybody. My name is Ken Arnold, and I am Director of Medical Museum. A quick plug for anyone who's not visited us, please do come to the museum. I'm also head of Program 4 here, and our interest is in putting cardiometabolic research in a historical, cultural, social context. And it's my enormous pleasure to introduce Vanessa Heggie, who's our next speaker. Vanessa is associate professor at the University of Birmingham, is a historian of science and medicine, particularly focusing on 19th century life sciences, biomedicine. She's published very widely on the history of sports science. In fact, we were just there with Leslie and Julene, on Victorian nursing, and on exploration and physiology. She's also been awarded an enviable number of fellowships and grants, immediately putting herself in a rather endangered species these days of a well-supported humanities scholar. And this afternoon, she's going to talk to us about the awkward history of trying to do metabolic research in extreme environments. She's given herself the playful title of extremely boring. I imagine within a sentence or two, we will know that that isn't true at all. But over to you, Leslie.
Thank you. Okay. So I'm not going to spend too much time trying to introduce my work. I'm going to give you 3 historical case studies, and I think they explain the sort of history that I do better than me trying to explain it to you. I am going to offer you a caveat up front, though, that despite the picture, I'm not an explorer, I'm not a scientist, I am in fact a historian. And the studies I'm going to talk to you about are very, very basic. There's no genes, there's no proteins, there's virtually no graphs or tables or anything like that, so lower your expectations for these, okay? So I'm also not going to show you any pictures just to start with, because I want you to try and visualize in your heads the experiment I'm going to explain to you and see what you think it must have looked like. So I'm going to take you back to 1911. So it's more than 100 years ago. And 3 British men are about to take part in a very exciting physiological experiment to discover how the human body metabolizes food when it's doing extreme exercise. This is a very basic experiment, and that is because our understanding of nutritional science at that point is indeed very basic. So what they're trying to work out is how the core food groups, fat, protein, and carbohydrates, work when you're doing hard exercise. What's the right ratio of those to help you do lots of physical work? So through the second half of the 19th century, there'd been a debate about this. The famous German scientist Justus von Liebig had stated in the 1840s that it's only protein that fuels the body. So muscles are broken down when you do exercise, and they're rebuilt when you rest. And a variety of scientists have sort of opposed this and tried different experiments. So that's what these 3 guys are trying to solve. These British men are therefore offering themselves as guinea pigs to this unique experiment that no one's ever tried before. They're using very basic foods. So they're using butter for fat, they're using biscuits for carbohydrate, and a kind of beef protein powder for the protein, and tea, because obviously they're British, they have to have tea. And basically what they're doing is that they start with a ration that's very high in one of those food factors. So there's a doctor involved in it, he's guiding the experiment, and he gets the high-fat diet. They recruit someone from the military, a sailor, and he gets the high-protein diet. And then they also involve a gentleman, a man so wealthy he doesn't have to have a job, and he gets the high-carbohydrate diet. And then they take part in intense, mostly outdoor physical exercise, not just for a day or 2, but in fact for weeks. Again, basic experiment, no lab-based chemical analysis or anything like that, and it's about self-reported experiences. So what the men were doing under the direction of the doctor was being reflexive about how their bodies were working. How hungry were they feeling? How well were they able to do the work that they needed to do? So, for example, the doctor on the high-fat diet and the sailor on the high-protein diet both found it very difficult to consume enough calories to do the work they needed to do. Like, basically gorging on butter is actually quite hard, it turns out. But the high carbohydrate guy, although he got enough calories, he felt hungrier and he had cravings for fat. And it turned out he had some other problems too, like poor circulation in his fingers. He actually got frostbite at one point. So over the days, these 3 men adjust their rations bit by bit. They're paying attention to how it makes them feel and how they're performing. And the big finding here is that all 3 of them, despite being quite different guys, they all come to the same ratio, which is more carbohydrate, but not exclusively carbohydrate. And it's roughly 4 parts of carbs to about 3 parts of protein to 1 part of fat is what all 3 of them agreed was the best possible ratio for them to do the hard work. Now, I've said it's a rough experiment, and it is, but it's also vigorously controlled. Unlike a lot of dietary experiments, these men have no access to any other food, and the food they do have has been weighed and measured, and it's understood how much they've got and how much they have left at the end of the experiment. The men bunk together. They're in each other's company for almost 24 hours a day, so they're able to reflexively sort of correct each other's statements. Like they could say, oh, you were working hard that day, or you weren't working that well, or you didn't finish your dinner on this particular experimental day. And although they're doing the exercise outdoors and are therefore affected by the weather, they are keeping a record of the weather conditions, and obviously they're all affected in the same way. They're all experiencing the same environment. So, even despite all these challenges, they come to this really interesting conclusion, a very solid conclusion about what could be the right ratio in a diet for you to do hard exercise. And yet, you will never, ever find out about this experiment from any journal. It's not published in any journal of physiology or nutrition. It's not a short note in the BMJ or anything like that. You can't find out about it in textbooks. So why not? Okay? The only place you can find out about this particular experiment is actually in someone's autobiography that was published in 1922. So that's a decade after the experiment took place. So why? Why did it take so long? Why is it so hard to find out about it? Now, I don't know what you were imagining, but this is what's going on. 9 months after this study took place, 2 of the participants, the doctor on the high-fat diet and the sailor on the high-protein diet, were dead. And the third of them, the gentleman on the high carbs, was traumatized by the experience of finding his dead colleagues. This is because they are Dr. Edward Wilson, Henry Bowers, and Apsley Cherry Garrard. They're members of Robert Falcon Scott's ill-fated Terra Nova expedition, during which the South Pole Party all died on their attempt to return from the pole. Now, Scott had asked them to do this experiment. The food they'd been testing, which is pictured above here, is Antarctic biscuit, pemmican, and butter. And they had done this experiment during the worst journey in the world, as Cherry Garrard later called it, which is a polar winter journey to collect penguin eggs from the emperor penguin's breeding grounds in Cape Crozier. Now, years and years ago, when I first discovered this story as a postdoctoral researcher, fresh out my PhD, I told it to a much older physiologist who I sat next to at a conference. And he said, very nice story, but it's of no relevance at all to the history of physiology, because they never published it, so it didn't have any impact on science. I'm not saying I've spent the rest of my career trying to prove him wrong, but it was very motivating. Because as a historian, I knew that this mattered, and I obviously had to find a better way of trying to communicate that to people. Now, I think anyone who thinks about it knows that you can learn a lot about science from work that wasn't published. For example, the fact that these men did this experiment, this challenging, detailed experiment, while also going through this intense, life-threatening experience of trying to collect emperor penguin eggs, right? That says they must have thought this was important knowledge, right? This was a really important scientific question that they had to solve. And of course, a huge amount of scientific knowledge, especially when it comes to experimental practices, is never actually published. Instead, it's shared in places like this, in conferences, by word of mouth. You're having trouble with that particular centrifuge, or this is how I've got it to work, or I've heard someone is working on this particular thing, or you might want to use these particular reagents, and so on. Those sorts of discussions happen in spaces like this. So I think future historians of science are going to have to spend quite a lot of their time combing through mailing lists and message boards and doing a lot of interviews to find this less formal part of the history of science. In some ways, my job is a bit easier because a lot of my resources are handwritten letters between the scientists that they have very meticulously kept, unlike emails, which we tend to just delete, or better yet, they are printed resources. Scientific explorers, particularly to spaces like Antarctica or Mount Everest, they didn't just read scientific papers, they also read the biographies and autobiographies of climbers. They went to talks by explorers at the Alpine Club. And information is also passed on in the field. So for most Western expeditions to Everest, the Tembochi Monastery pictured here is a very convenient stopping place. And climbers meet here, and scientists meet here, and they talk about the equipment they're taking. They talk about where they've left food caches on the mountainside. They talk about what routes look the most promising and tips and tricks for how to get the oxygen systems to work at altitude. And I'm trying to write, therefore, a history of science that isn't just a literary review of the great work, but that recognizes science as a human activity. And that often means having to try to reclaim and to reconsider fieldwork, grey literature, lost conversations in the corridor at a conference like this. So it's basically what's happening in spaces like this that I'm really interested in. And fieldwork in particular is quite under-considered by historians, at least when it comes to medicine and physiology in the 20th century. The major work on Antarctic science, which is this epic, A History of Antarctic Science by G. Fogg, published in 1992, so it might need a bit of an update now. This claims that prior to the Second World War, I'm going to quote him, there was little that could be dignified by the name of medical research in Antarctica. And I think that's a very unfair reading of the hard and careful work that was being done in 1911 for nutritional research. And so I've been trying to sort of reset the balance so that we realize how much work, a lot of it turning out to be physiology and indeed metabolism, has been done in these extreme environments in the 20th century, regardless of whether anyone actually published it or not. One of the reasons we don't know that much about early research work in extreme environments, which is Antarctica, the high Arctic, high altitude, but I also include desert research, is because a lot of it is actually shared in those more informal ways. Another reason why we don't know much as we should about these field experiments is also hinted at in the sarcastic title of my talk, which is extremely boring. So generally, when people read about expeditions to the South Pole or to the summit of Everest, they are expecting it to be exciting, even if it's a scientific expedition. They want drama, and they want risk, and they want overcoming obstacles and stories of triumph and tragedy. And so as a consequence, sometimes the science gets left out, and no offense to anyone in this room, because it can seem kind of boring compared to that stuff. Even today, there is a huge amount of repetitive and routine work involved in the practice of science. And it's not something we often talk about in terms of the history of science, but it's also not something we necessarily talk about when we're trying to recruit young people to come into the profession or to do science degrees. And I'm going to confess here that one of the reasons I've grown up to be a historian and not a scientist is because at 18, when I went to university, I was horrified by how much of a degree in genetics just involved pipetting like this over and over again, and I was looking for something else. Okay, so I guess my first example, which is surviving on frozen butter in Antarctic winter in the permanent dark and also having to fight emperor penguins who are like a meter tall and can stab you if they're trying to, with their beaks, if you're trying to take their eggs. That is pretty dramatic and exciting. So as a second example, I'm going to stay in Antarctica, but going to try and find something that has more boringness built into it. So we have moved on 50 years. It's the International Geophysical Year of 1957 to 58. Antarctica is teeming with geophysical scientists in particular, but also with explorers. And one exploration party is the one that's whose route's being described here. It's the Commonwealth Trans-Antarctic Expedition. It is a joint venture between the British director of the Falkland Islands Survey, Vivian Bunny Fuchs, pictured here on the left-hand side, and the famous New Zealand climber Edmund Hillary. That's not Hillary, that's actually a very young David Attenborough in the picture up there. So the plan here was that they were going to manhaul, that's physically drag sleds, from one side of the Antarctic continent to another. It was a record-breaking attempt. No one had ever done this before. They were also going to do a major human experiment into human responses to cold, metabolic and adaptational responses to cold. I think there's probably at least some people in this room who know a little bit about thermal research. If I say Antarctica, you might think of Paul Sippel, who was an Antarctic explorer and a scientist who coined the term wind chill factor in terms of sort of measuring human responses to cold back in sort of the 1930s. You probably won't think of Dr. Alan Rogers. And that's despite the fact that Rogers did some of the most long-term, extensive studies on human acclimatization to extreme cold that have ever been completed. So, Rogers' story. Rogers decided to take this opportunity to address a really pressing problem in physiology, which is, do human beings acclimatize to cold? Again, might seem like an incredibly basic question, but even in the 1950s, the physiology of cold adaptation was not clear, and it was highly disputed. There was evidence of changes in comfort sensations, there was evidence of some adaptation of the peripheries, but almost no evidence that, unlike heat or altitude, the human body meaningfully adapted to extreme cold. And that's partly because it's quite difficult ethically to expose people to extreme cold. And even in the Arctic or the Antarctic, people obviously wear clothes, and they generally live in heated buildings for most of the time. So what Rogers wanted to do was to study extreme exposure over a long period, and that's with the men who are pictured here, the members of the Trans-Antarctic Expedition. About a dozen men in the end became his subject group, and they're going to spend more than a year on the Antarctic ice, and they're going to be living in tents. And his study is very simple. He was going to make them keep a diary of how much clothing they wore, take weather and temperature measurements, allow for complicating factors like, are they doing a lot of exercise, do they feel ill, what have they been eating? And he's simply going to see if men who spend an extended period of time in the Antarctic wear less clothing at the end of the expedition than they do at the beginning. So it's a very beautifully simple research question. Do adult men achieve the same level of thermal comfort with less clothing after 14 months of exposure in the Antarctic desert? So this is an example of the data card that he used. You can see that it's very complex. It's not just the clothing, but it's also activities, sleeping patterns, things like that. There's actually space on the back for you to put extra comments in if you thought there was anything else that would be relevant. I think it's worth pointing out what enormous compliance Rogers got for this experiment. This card had to be filled in every day by every man for months of trekking through Antarctica with extra notes on the back. And amazingly, only one set of cards came back incompletely filled out. He had amazing compliance from the participants in the expedition. So he gets home with all of these cards. He's carefully packed and transported them across half the world from Antarctica to Bristol in the UK. And then he asked the professor of statistics to help him figure out the correlation and causation that's going on here. And the professor agrees, and within a year has dropped out. It's too hard, it's too complicated, can't do it. Rogers finds another professor of statistics who agrees to do it. Again, he starts. Within less than a year, the professor drops out. I can't do it. It's too much maths. It can't be done. One of the reasons this is so problematic is because clothing is very difficult to standardize. Rogers needed to find a way to convert the real clothing worn by actual human beings to a number that could be used in an equation. And I'm sorry about the quality of these photos, but they're taken directly from the reports. What he did was, for every individual man for whom he had the complete set of cards, so that's for 12 guys, he took their warmest and their coolest outfits, that's 24 different outfits in total. He either made a replica of them or he used the original clothing that the men had, and he sent them to the Wright-Patterson Air Force Base in America, where they were analysed using a thermal copper man that gave clo values for each outfit. And with the warmest and the coldest clo value, he could then extrapolate the in-between outfits they'd worn. So far, so good. He also, for double checking, sent 4 of these outfits to the copper man at the British Royal Air Force Base, the Institute of Aviation Medicine in Farnborough in the UK. And guess what? The British copper man gave completely different measurements for the 4 outfits that he sent to them. Thermal properties, as I'm sure many of you know, are hugely variable according to human action. Untucking a shirt, unzipping something ever so slightly can drastically alter its thermal potential, as can having it wet or frozen. Even something like whether or not a man has a beard can make a significant difference to how warm a scarf or a balaclava can be. All of this was too complicated for the statisticians, and that is why they gave up. The cards sat in a file. Rogers couldn't do anything with them. All that data, all that effort, all those human subjects who'd conscientiously been filling in a card whilst trying to also go across Antarctica, apparently for nothing. And then in 1969, he gets his big break. He is presenting at a conference, because we know that's where all the important things happen, and the American military expresses interest. He gets a grant from the US Air Force, and it's enough for him to hire an analyst. He hires Mrs. Rosamond J. Sutherland. She's a mathematics graduate, but most importantly, she is a computer programmer. And she is actually able to write a computer program that can analyze over 1 million bits of data, which is a massive task for a late 1960s computer the size of a room. They also had enough money to hire 2 other women as administrative assistants, Mrs. E. Fountains and Miss Bean Thornton. And Thornton's role was crucial because she had to code this clothing data. She had to turn these messy cards of tick boxes and outfits into a numerical input that could be turned into a punch card that Sutherland could then put into the computer. So finally, they did it. As you can see from this picture, the final report of the heat acclimatization experiment, or cold acclimatization experiment, was published in 1971. We have seen some lags in publication data here, but I think this is the biggest one so far. So from data collection in 57 and 58 to final publication in 1971. And what did they find? Nothing. It's null. There's no evidence of significant human acclimatization to cold, it turns out, with the possible exception, as we'd expected, of small comfort changes to the face and to the hands and maybe the feet. I'm sure many of you know it is much harder to get articles accepted if they prove a null hypothesis or if they are successful replications of work that other people have done. And I think it's probably very lucky for Rogers and Sutherland that they got this military funding because it required them to write a report that was then physically published and put into military libraries. Okay? They might have found it much, much harder to get this published in an actual journal article. In fact, the rest of us had to wait another 7 years to get this material. Rogers and Sutherland did finally get a mainstream publication in 1978, so 20 years after the data was collected, and even then it's not a full article, it's like a short note of a couple of paragraphs in the Journal of Physiology. Null results really matter. After all, the core part of the scientific process is supposed to be independent replication results. And if we have a system that doesn't support and publish such practices, we are in real trouble. But more than that, what's lost in the story of Rogers and the Transantarctic expedition is actually he did discover some new things as well. It wasn't just repeating or reaffirming what was believed. So for example, Rogers and Sutherland's work was also used to correlate between Clos values and the number of layers. So in many much earlier, older studies of thermal metabolism, Clos values are not used. Instead, the scientists just use the number of layers of clothing as a proxy for the insulation. And therefore, if you want to be able to use that earlier data to compare to more recent ones that have Clos in, you need that way of measuring between the two, turning layers into Clos values. So this is a really useful methodological tool that just kind of vanished into the ether. More than that too, right from the very start, Rogers and the rest of the Transantarctic team found that we were wrong about a lot of what we thought about how clothing worked in extreme cold. Crucially, Rogers found from his own experience and from anecdotes, and then Sutherland confirmed it with her statistical analysis too, that the traditional laboratory and climate chamber-based thermal studies paid way too much attention to wind chill as a factor and nowhere near enough to the solar radiation reflecting back off the snow. And in fact, repeatedly, the participants in the Transantarctic expedition found they were too hot in the recommended outfits, particularly when they were doing heavy work like man-hauling. So the story of Rogers, who, I should remind you, is an Antarctic explorer, he put his life at risk to gather this data, and Sutherland, who was a woman who was pioneering in a field that was deeply undervalued, and as soon as it did become valued, it became male-dominated. That's another story about computing. But their story illustrates for me several of the key points about the history of science that I try to write. Firstly is the obvious one that fieldwork really matters to the history of science, particularly in medicine and physiology. At the moment, the 20th century is still written about and quite often taught as the era of big science, the rise of the laboratory and so on, and that's only a partial story, and we need to put these sorts of stories back into it. But secondly, scientific fieldwork is easily lost or its results can be hidden. It's hard and it's also very expensive. And that means you're often doing 2 or 3 things on an expedition. You're not just doing a biomedical expedition, you're also trying to do geology at the same time. Or you're not just doing a physiological experiment, you're also trying to do a world-breaking attempt to cross the Antarctic desert. And that means that the story of the science can often get lost in the story of the exploration itself, or the less successful bit of the science kind of get buried or drowned out by the more dramatic stories of discovery. Particularly if most of the story is, waiting around for a computer to be invented so that you can actually analyze your data, hence the idea of being boring. Okay, so thirdly, I think a lot of scientific fieldwork influences through grey literature, through word of mouth, through informal routes. We know that if you get a null result, it can be hard to publish, so perhaps there have to be other ways for you to share this. And quite crucially, in many cases, the funding of an expedition doesn't actually stretch to the analysing of the results of the expedition. Again, this is a boring economic point, I guess. But when you look at the outcome of a lot of scientific expeditions, including those that collect physical samples like penguin eggs or rocks or things like that, the failure to produce impact is often because of the lack of funding or interest or support after the expedition itself. And I think that's fairly obviously an important point for funders now about the longevity of projects and what we expect them to be. Now, Rogers' example might seem relatively extraordinary, waiting more than a decade for the right money and the right computing technology, but it is not that unlikely that in a decade a new technology will appear that will totally revolutionize the analysis of already collected data. But you need money to store these items safely, you need money to keep people working on the projects and so on. So that follow-on funding is very obvious when you look at expeditions and is a crucial point, I think, for everybody's project. The other big area where perhaps more informal communication really matters is in methodology. So a lot of what you do as scientists never makes it into the methodology section of the stuff that you publish. So for example, if you wanted to replicate the nutrition experiment of 1911, you need to know the recipe for the pemmican that they used. You need to know how to get a license to go to Antarctica. You need to know how to put up a tent in the middle of Antarctic winter. You need to know how to collect a penguin egg from an emperor penguin and not have it peck you to death, and so on. So even if Scott and his team had survived and come back and published his research, that sort of information would never have been included in the journal article itself. That's what's passed on in the corridors, in the informal spaces. There is a whole world of scientific practice out there that isn't captured in articles or conference papers or textbooks, and that is what a historian like me really searches for. So I'm going to wrap up. I want to tell you a final story. And that's the one where the results of the fieldwork did get published, and they got published really quickly in a big journal, and they still got forgotten and ignored. And partly I'm putting this story in because we've only really been in the Antarctic so far, and that's 1911 and 1958, and I want to take us to a different extreme environment, which is Everest, in the middle of those two. So this is Everest in 1924. So this pictured here is Theodore Howard Somervell. He's a doctor and a surgeon. He went on two British Everest expeditions in 1922 and 1924, and then went on to be a medical missionary across India. His expeditions definitely had drama. In 1922, 7 of the local Sherpa porters were killed in an avalanche, largely due to some of the choices made by the expeditionary team. These are the first recorded deaths on Everest. And then in 1924, the first European deaths, as George Mallory and Andrew Irving disappear somewhere near the summit of Everest. I also have this nude photo up, because I often see this photograph as being credited as being George Mallory. Mallory has a very strong queer identity. Historians have written a lot about his sexuality, his sexualized body. When I found the original of this photo, it was very clearly marked with the initials THS at the bottom, so I think this is in fact Howard Somervell, so I'm just putting it up there to reclaim him from the shadow of his more famous mountaineering partners. Likewise, not many people know that Somervell himself actually made an attempt on the summit in 1924. He was roped together with the deputy expedition leader, Edward Norton, and he had had a really bad sore throat for days at this point. And in the last 1,000 feet or so on Everest, he started to cough and to cough and to cough, and suddenly there was something in his throat and he could not get his own breath. In his own words, he couldn't get Norton's attention, and he literally sat down in the snow on the mountain thinking, this is it, I am going to die. But the last-ditch attempt, he did a self-Heimlich and managed to cough something up. And he survived, and it turned out that he had frostbitten the membrane lining of his larynx, hence the pain. It had, the cells had died, they'd sloughed off, and he was basically choking to death on his own dead skin cells. So that horrific story is relevant because Somervell's research project on Everest was mostly respiratory. Okay, so we've done cold research, we've done nutritional research. I'm going to finally very quickly have some respiratory research and also some blood research. So he had borrowed a gas analyzer from the Medical College in Calcutta on the way into Everest, and he was collecting respired air from the other mountaineers using the inside bladders of footballs, soccer balls. And he used these to gather gas from the mountaineers and then take it down to base camp and do an analysis of it. And this research he did publish. He published very quickly a short note with tables in the Journal of Physiology in 1925. Actually, his work wasn't that great because the inside of soccer balls turns out to be permeable to CO2, carbon dioxide, so the results were a bit thrown off. But actually, that's not the bit I'm interested in. The bit I'm interested in is something else he said that got ignored, which is this paragraph. It's literally the last paragraph of the article. It finishes exactly after this. It's almost a throwaway comment. And here he is doing two brand new extraordinary things. Firstly, he is testing Sherpa blood at altitude. No one had ever done this before, and no one would do it again for another 40 years. Secondly, the finding, in case it's not clear from you from the old-fashioned language, is of a low red blood cell count. So rather than finding packed blood, polycythemia, which is an increase in red blood cell production in response to altitude, he's finding the opposite. This discovery contradicted every single finding on altitude and blood ever made up until this point in 1924. It is assumed in 1924-25 that every human being responds to altitude by increasing red blood cell production, and we don't. This is revelatory, but it's going to be completely ignored by all the altitude blood researchers for at least the next 40 years. I don't have time today to completely explain why. The history of blood research altitude goes back to Mexico in the 1850s, forward into Peru into the 1950s, but in the early 20th century involves a lot of the names that might be familiar to you. I've put Ansel Keys up here, but also Haldane, Barcroft, all that sort of people. Work across the Americas, in North, but mostly in South America, and also in the Alps, had shown that everyone at altitude has polycythemia, pronounced in permanent residents, gradually increasing in visitors. And these people extrapolated their results from South America and declared this a universal human trait. Everybody at altitude has a body like this. Why did they extrapolate like that? And unfortunately, my historian's answer is race science. In amongst these studies, and these scientists' writing, you find a very strong sense of environmental determinism. Certain sorts of environment make certain sorts of bodies, make certain sorts of people. All people at altitude are the same. It's a homogeneous racial category. We, of course, know that's wrong. There's a range of human responses, both evolutionary and adaptational, to altitude. Only one of those is increasing red blood cell production. But a real commitment to this understanding of homogenized, essentialist, racialized understanding of altitude physiology is the only really strong explanation I can come up with why Somerville's findings didn't have more impact. altitude physiologists, high altitude mountaineers in the 30s, 40s, and 50s were a very small exclusive group. They all went to the same universities, they all went to the same men's clubs, they knew each other, they cited each other's work. Doctors to Everest always went back to previous doctors on previous expeditions and asked them for advice and for guidance. So I can't believe that they somehow didn't know that he'd done this work. I think it's also notable that the only other people who were doing blood work in the Himalaya rather than in the Andes were the Nazi-sponsored expeditions in the 1930s, who obviously had their own ideological commitments. And all that says to me is that there was a stronger ideological reason why the sameness of altitude man was a really important part of altitude science until the 1950s, despite some of L's clear contradictory findings. So, failed research, uncited research, unpublished research, boring research, unpopular research tells us massive stories about science. And it can draw our attention to dangerous blind spots, whether that can be forms of racism or that we desperately need to have proper follow-on funding. Thank you so much for your attention.
Thank you so much, Vanessa. So we have just a few minutes for burning questions. I will jump in then, but please, if you're almost there asking a question, follow me immediately. My question for you would be, we've got a fantastic range of scientists here. What would you advise them to do in order to make your historian successors in 10, 20, 30 years' time find untold riches in the stuff that might otherwise get hidden?
I mean, one is don't delete your emails, please keep them somewhere. We've got to find a way of keeping some of this digital information because that's the main thing we lost. But actually, I think it's more about having conversations across the university. I know we talk a lot about interdisciplinary study and so on, but it's one of those things that's much easier talked about than it is necessarily done. And I think realistically, talk to the historians need to talk to scientists, but the scientists also need to bring the historians in. And some of it is, you've got to make sure that we still exist. I don't know what the situation's like in Denmark, but history departments in the UK are closing. Like, we are losing our funding. Our major funding bodies have already drastically reduced the amount of money they put out. And there's a certain feeling like if we don't have the protection from the better funded areas in STEM, there won't be historians to do this work in the future. So I think some of it is just show a little bit of love to the people in the humanities, because often they are really interested in learning from what you're talking about and what you're doing. It's such a human activity. If we don't know you as human beings, it's really difficult for us to write this sort of work. So keep your staff, make more friends.
I see two hands, one there, and then we've got a second one here.
Hi, I'm just here. Ben Stocks, CBMR. Former sports science was from the University of Birmingham, actually. So, hey. We are always doing lots of cutting-edge research, but if we're honest with ourselves, often we're probably reproducing old findings that we didn't discover in the literature before we'd done our experiment. Have you got, as someone that is searching for old knowledge a lot, have you got any advice to us guys?
I mean, firstly, you're not doing any worse than they did in the past either, because I think it's interesting that as we've got more, as searching has become easier and we've got more digital resources and it's easier to do the literature searching and things like that, the amount of it's become so vast, it's actually uncontainable. And the fact that it's very clear when I'm reading the archives of the people in the 30s and 40s, even though there are only 10 people doing altitude science at this point, they still sometimes did miss stuff. So don't feel bad. This is what's happened all along, okay? But again, I think the second thing is that would be the moral out of this paper, is that so much of this stuff is communicated by word of mouth. And in some ways, I don't want to replicate that because it's an act of privilege that certain people's science gets cited and then everybody knows that person and that's a citation classic. So I think it's partly about, it's about expanding your friend group. It's about getting to know more people, talking across the divides of the disciplines and reading that sort of work and coming occasionally to a conference that isn't in your main field and seeing what's going on. So I think it's a lot of that is about making sure that you have a really diverse group and then acknowledging that actually a lot of this work is not done when you're sitting searching a database. It's done when you're in the coffee room or when you're in a corridor and things like that.
And then a question here.
Yeah, Vanessa, that was absolutely fantastic. Thoroughly enjoyed that. Thank you so much. I wanted to ask you about how you see social media playing into this in the future. I was thinking about some of the discussions that happen on Twitter, and how do you see that historians looking back on social media and using that for inference?
Yeah. Well, one of the things that makes me feel really old is when my current students are already using social media, because it's like, that was 10 years ago. Can I use that in my history dissertation? I'm like, I guess you can, but it's making me feel like a riveled prune. But Yeah, the real challenge here is about data storage, and that's a little bit why I put the mention in. Like, there has been some big projects, many national projects for sort of archiving the web and so on. The British Library has a huge project about collating public tweets, at least, and trying to save them. But the reality is it's incredibly expensive to store all of this data, and it also becomes inaccessible. Like, there's a whole load of floppy disks and punch cards and things like that where people have wanted to reclaim the data, and they've had to basically go around and find computing nerds who've got one in their own basement out of their toy copy out of their sort of hobby, because we're not keeping the equipment to read the data sources that we've got stuff on. And some of this is about getting good curated. Like, I think probably every science institute like this needs at least one curator, if not a curator and a museum expert and an in-house historian.
Oh, several, I'd say.
It's like, a handful, about six, would that do? But you need those.
For a start, yeah.
But it just in terms of like the data conservation, those are the people who in 10 years' time will have the skills for the things that 20 years old, which is also partly when you want to get access to the historical records, it might not be a published paper. It might be the box folder of punch cards that no one ever processed, and that's what you really need. And I think that's where, in a practical sense rather than in a sort of intellectual sense, having a historian who's actually embedded in the institute itself is incredibly helpful, because if they can't do it, they'll be able to find someone who can.
Okay, great. I think, unfortunately, you'll find each other and you'll ask your question directly. We should stop there. I think I've got three things left to do. One is to remind you to be back here for Rachel Freethy's talk at 3:45. My second thing is to tell you now to go and hunt out the posters you've not already seen and have richer conversations there. And third, and most important, is to ask us to thank Vanessa again, and indeed for me to pass on a small gift.
Thank you.